Welcome to Zakaria lecturing. Our course we continue. Today we start lesson 3 in microbiology. Our previous lesson we tell two theory of abiogenesis and theory biogenesis. Today we start abiogenesis theory. Theory of abiogenesis deals with the theory of spontaneous generation, stating that living things originated spontaneously from non-living things. Aristotle 322 BC, the founder of the theory of spontaneous generation. He observed spontaneous existence of fish from drying bone when the bone was filled with the rain. Abiogenesis states that the life comes from pre-existing life. Francisco Redi 1697. He was the scientist who first tried such experiment to disprove spontaneous generation. He took these two experiments. He took the meat and exposed it, uncovered it. And then after he see maggots are when something decay produce maggots. And other experiment he covered the meat, and then the result there is no maggots. Francisco Redi introduced experimental procedure to disprove spontaneous generation. Performing experiments that disprove theory of spontaneous generation for more complex forms of life began approximately 1686. Utilized jars containing meat, some were covered and some were not. Maggots appeared in uncovered jars and concluded that maggots did not emerge spontaneously but from the eggs laid on the meat by the fly. The controversy, the means the quarrels on spontaneous generation took 200 years. Louis Pasteur, 1895, was scientists who disproved the theory of abiogenesis once and for all. He was eliminated this theory. Performing experiments to disprove theory of spontaneous generation, he designed large curved flash, swan necked, Pasteur goose neck flash, and placed sterile infusion broth, meaning solution. Flash remaining sterile unless tilted or neck broken. In air freely moved through the tube, but dust particles were trapped in the curved portion of the flash. No microbial growth was observed. This experience partition A, he heated this infusion, then steam escaped from opening of the flash out. Infusion cities, no microbes appear months after months. Air moves in and out to the flash. Infusion remains indefinitely sterile because there is dust particles settle in the bending. Portion P, sterile infusion sits in flash, and this is dust particles. When tilted, the flash is tilted, sterile infusion comes in contact with the dust. Microbes appear in infusion after solution or infusion attach or contact with dust. Therefore, Pasteur proved that microorganisms enter into the broth with their air, and microorganisms do not evolve spontaneously. Other major contribution of Louis Pasteur, he produced microbial theory of fermentation. principles and practices of sterilization and pasteurization, control of disease of silkworm, development of vaccines against anthrax and rabies, discovery of streptococci bacteria, germ theory of disease. Pauster has also developed the germ theory of disease, which states that a specific disease is caused by a specific type of microorganisms. Report occurred in 1867, established an experimental procedure to prove germ theory of disease, which states that specific disease is caused by specific pathogen. The scientific procedure is known as Koch's postulate. Koch's postulate proved the germ theory of disease. Microorganisms can be accepted as causative agent of infection disease only if the following conditions are satisfied. The microorganisms should be found in every case of the disease and other conditions which explain the pathological changes and chemical changes. It should be possible to isolate the causative agent in the pure culture from the lesion diseased portion in the body. When such pure culture is inoculated into the appropriate lab animal, the lesion of the disease should be reproduced. Point 4. It should be possible to re-isolate the bacterium in pure culture from the lesion produced in an experimental animal. Koch's postulate this experiment, these 4 points. He takes sample diseased animal and then he culture it in artificial media. Then artificial media he take sample and inject in healthy laboratory animal. And then he take sample after injecting. Then he culture. Then first culture and second culture are the same.